And that better?
That's better. Thank you.
Perfect. Okay. Researchers studying relationships between phenotypes often use phylogenetic comparative methods. So the goal of these methods is to look for pairwise correlations between phenotypes of interest. We specifically model the phylogenetic relationships between all of the taxa of interest to avoid spurious correlations and things like that. It's also, it allows us to map things back to the phylogeny, which can be useful. The challenge of these comparative methods is that they don't scale well to big P problems. So those are problems where you have lots of different phenotypes that you're trying to understand the relationships between. One problem we were interested in looking at, you can see on the, trying to get access to the, here, anyway, on the left here is looking at the domestication of yeast. So there were 82 different phenotypes related to environment stress, nutrient stress, their ability to sexually reproduce. In these phylogenetic models, 82 traits is actually a lot, and it really, this is a little bit too big for those to handle easily for computational reasons. Additionally, if you want to study high dimensional phenotypes, for example, we have brain shape here. You can't have a single measurement or single observation that captures brain shape. And so typically you need to rely on some kind of dimension reduction to, model or at least map this back to a phylogeny. The solution to this problem is what is known as phylogenetic factor analysis, or at least that's one solution to this problem. This was developed by one of Mark's former graduate students, Max Tokov. Basically, it's like a normal latent factor model, except that we have this phylogeny over there on the left. We've got our phylogeny, and we have our p-dimensional data, where p is some big number. And what we do is we assume that the evolutionary process on the tree is some low-dimensional evolutionary process. So that's going to be a k-dimensional process, where k is something relatively small, 2, 3, maybe 10. These k-dimensional factors evolve independently along this phylogeny. And what we need to do is map this low-dimensional process to the high-dimensional process. We do this via a multiplication by a loadings matrix L, which we need to infer. And then we add some Gaussian noise, and that gets us to our data. So this is kind of the simple version of the statistical model. Now, the big challenge with these phylogenetic models generally is that the latent factors, those Fs, they are not independent. The evolutionary history, the phylogeny, induces statistical dependence between these factors, and that can create computational bottlenecks when trying to do inference on these models, inferring both the factors themselves as well as that loadings matrix, which is a parameter of interest. So the way we get around this is we use a common strategy in these phylogenetic models, or really any kind of directed acyclic, or models that rely on directed acyclic graphs. Basically we start with our phylogeny here over on the left, starting at the root E and moving toward the tips A, B, and C. And we pull this phylogeny apart into these pairs of ancestral states and the tips. If we condition on the ancestral states, in this case, E and D, then all of our data are now independent. That's much more easy to do things like compute likelihoods with. Now, the trick is we don't actually observe any of these internal states. So we have not observed the factors at D. We have not observed the factors at the root E. What we need to do is compute these independent likelihoods conditional on these internal nodes. And then as we reassemble the tree, we need to integrate out those unobserved states. And because this is a Gaussian model, we can do those integrals analytically. without very much computational cost. And so as we reassemble the tree, we propagate information from the tips of the tree back to the root, and we can use that general approach to compute our likelihood and really any other or most other statistics we need for the inference. And this scales linearly in the number of taxa, which is really nice. To give you an example of some of the analyses that we are working on here, this is still work in progress, but we're looking at the domestication of industrial yeast. So yeast used to brew beer, make wine, bread, ethanol, things like that. And so we did what's called an exploratory factor analysis, where we just tried to identify, how many factors are there, and what traits are those factors associated with, and found that there were two factors, one of which was a dominant factor, explained the majority of the variance, and that factor was very clearly associated with what we are labeling a domestication signal. So that factor was associated with lower tolerance to nutrient stress, lower tolerance to environmental stress, as well as a decreased ability to sexually reproduce. And so when we map that factor onto the phylogeny, we see a very, very clear pattern with that kind of yellow clade on the right, showing a high domestication signal, and the rest of the phylogeny showing a relatively low domestication signal. And this right clade, that domesticated clade, that's mostly American and British ale yeasts, whereas the rest of it correspond to all sorts of applications. But you can actually see towards the left, there's another beer clade, and those are mostly the Belgian beers. And so there's actually there's reasons to expect that those would be, quote, less domesticated, due to kind of the specific nature of how the ales versus those other beers are produced. So that was kind of interesting to see. So moving on, one, again, super preliminary work we're doing as well is you can actually treat individual mutations as phenotypes of interest. So you can map these kind of binary 0-1 mutations to some continuous space. And you can actually look for now interactions between genes themselves, as well as between those genes and quantities of clinical interest or other interest. And so with this HIV data, we looked at 20 mutations, ran a factor analysis on it, and one of the factors we identified showed a very strong interaction between this T186 mutation and that T190 mutation. And that's actually a known interaction. T186 is an HLA escape mutation that, in the absence of T190, reduces replicative capacity. But when you combine it with that T190 mutation, the replicative capacity is restored, and it then can escape the immune system better. So this is, I think, something I'm excited to explore, is using this to look for interactions between genes and how those interactions actually affect quantities of interest. We still have a couple of unresolved problems, one related to the loadings identifiability, where the loadings matrix is rotationally invariant with respect to the likelihood. That's a problem common to factor analysis generally, but one that we're trying to come up with kind of a slightly different approach to. And then also before, with that HIV data, we had binary data that we were trying to map to this continuous process. That's been done before in this phylogenetic context, but the factor model adds kind of a little extra nugget of stuff that we have to deal with there. So I'd like to thank my collaborators on this, as well as everyone else in my advisor group, as well as my advisor, Mark Suchard, and of course, my funding sources. So yeah, please let me know if you have any questions.